So this is week 6 and your first question is glutamate receptors are present on neurons, astrocytes, oligodendrocytes or all of the above. Glutamate receptors are present on neurons, astrocytes, oligodendrocytes or all of the above. Your time starts now. Start writing in the chat box. Then we'll discuss the correct answer. Is the slide not visible? I think it is visible. Okay, okay. Okay, okay, it is visible. Good. Start writing the answers. Glutamate receptors are present on neurons, astrocytes, oligodendrocytes, or all of the above. I need answers from all of you. So this week we are discussing about receptors, basically chemical messengers and receptors. Last week we discussed about membrane potential. This week we are discussing about receptors on the membrane, synaptic membrane. Okay, is everyone done? I am not able to see as many answers as students are here. It's okay even if you give the wrong answer, but please try to be active. Try to give answer. It's okay if you're guessing it, it's okay. Glutamate receptors are present on neurons, astrocytes, oligodendrocytes, or all of the above. Okay, we'll not waste much time. So the correct answer is all of the above. Okay, so glutamate receptors are present on neurons as well as in astrocytes as well as in oligodendrocytes. So the answer is all of the above. So here you can see, so what is glutamate receptor? A receptor that accepts that glutamate binds to that receptor. So what is glutamate first of all? So glutamate is an excitatory amino acid. It is a neurotransmitter, excitatory neurotransmitter, and it is one of the most common signaling molecules used in synaptic transmission. So glutamate receptors can be of two types. One is the ionotropic glutamate receptor and the metabotropic glutamate receptor. So what is the main difference between these two kind of glutamate receptors? One is that the ionotropic glutamate receptor has these ligand gated ion channels. That's why it is called as ionotropic glutamate receptors. Whereas these metabotropic glutamate receptors bind to the glutamate through a secondary messenger system. I think all of you must have read cell signaling. There are types of ligand gated receptors and G protein coupled receptors. Like that, here glutamate receptors are divided into two types. Okay, we'll discuss more about it in the upcoming questions. So we'll move on to the second question. NMDA is a glutamate receptor, GABAergic receptor, cholinergic receptor, a cholinergic neurons, or none of the above. NMDA is a glutamate receptor, GABAergic receptor, cholinergic neurons, or none of the above. Start answering. What is NMDA? I need answers from all of you. I can see everyone is not answering. This interactive session is only effective when you guys interact, right? When you guys give the answers. Okay, I can see some answers now. Nice. Okay, some are saying glutamate receptors, some are saying GABAergic receptors. Okay, most of you are saying glutamate receptors. Okay, let's see if it is the correct answer. Yes, glutamate receptor is the correct answer. NMDA is a glutamate receptor. We'll see how. Here you can see the structure of NMDA receptor. So structure wise they're very complex, as you can see, so many ions coming and binding to this receptor. Like it has separate binding sites for glutamate. This NMDA receptor has binding sites for different molecules like glutamate, like different molecules which act as neurotransmitters like glutamate, glycine, Mg2 plus, zinc, Zn2 plus, polyamines, etc. And these NMDA gated channels are more permeable to calcium ions than to sodium or potassium ions. We have already discussed about sodium and potassium ions and role of sodium and potassium ion in maintaining the membrane potential, but we have not discussed about calcium ions. So here calcium ions are also necessary, and this NMDA receptor is more permeable to calcium ions than to sodium and potassium ions. So this NMDA receptor is a type of glutamate receptor which is having a ligand gated channel, and its opening depends on both the agonist, that is the activating ligands, and the membrane voltage, right, which is maintained by the influx and efflux of the sodium and potassium ions, right? And here I've given a lot of notes about the NMDA receptor. You can go through these notes after the class. Um, Obviously, you will be getting these PPTs from the folder. You can learn from that. Okay. We'll move on to the next question because it's already late. AMPA is a glutamate receptor, GABAergic receptor, cholinergic neurons, or none of the above. AMPA is a glutamate receptor, GABAergic receptor, cholinergic neurons, or none of the above. The time starts now. Start answering. AMPA. Yeah, I also want every one of you to note down how many correct answers and how many wrong answers you're giving, and we'll see your score out of 10 at the end of this session. Okay? I want answers from everyone. Glutamate receptors, is it? Okay, let's see if it is glutamate receptor. Yes, AMPA is also a glutamate receptor like NMDA, but it is not similar to NMDA receptor. So AMPA is an example of a non-NMDA receptor. These receptors do not have ligand gated ion channels, and they are not more permeable to calcium. The difference is those NMDA receptors were more permeable to calcium, whereas these channels are more permeable to sodium and potassium. So these NMDA receptors are selectively agonized. Agonized means are selectively activated by a kainate. You can see here the kainate, AMPA or quisquilate. These are the type of ligands or molecules, you can say, which bind to these receptors and activate its function. So these associated ion channels are more permeable to the sodium and potassium ions. Here I have just mentioned all the full forms of AMPA and similar molecules which act as antagonists. Okay, we'll move on to the next question. LTP stands for LTP stands for last terminal potential, long-term potentiation, both A and B, or none of the above? These are very simple questions actually if you know the correct answer. LTP stands for last terminal potential, long-term potentiation, both A and B, or none of the above. Your time starts now. Start answering. If you have attended the lectures, I think these are very straightforward questions and you can definitely answer it correctly. These are not very complicated or twisted type of questions at all. And I think your present year assignments are also are also having similar kind of questions, and solving these questions will definitely help you to answer those questions correctly as well. Okay, is everyone saying B, long-term potentiation? Is it the right answer? I want answers from each one of you. Everyone be active and answer the question. It's okay if it's wrong or right. Either one is okay. Yeah, and obviously I'm recording these sessions. If you want to go through the slide, if any point I have not discussed and you want to go through it, you can uh, follow my YouTube channel and see the recording sessions there. If you have any doubt with the concept or you want to see or learn the questions again. I think I've given the YouTube link before, right? Okay, is everyone done with the answer? Should we proceed? Okay, let's see the correct answer. Correct answer is long-term potentiation. Okay, I think everyone has given the correct answer. So what is long-term potentiation? It is nothing but the strengthening of the synapses, right? The synapses which are involved in the synaptic transmission of the chemical messengers throughout between the two neurons, the strengthening of the synapses so that the chemical transmission can happen properly. And the glutamate receptors play a very major role in this long-term potentiation, that is strengthening of the synapses. Here you can see a normal condition with a synaptic transmission going on here. And what happens when there is more synaptic strength? You can see that more number of chemical messengers are getting transmitted through the synaptic vesicles, right? Here the chemical messengers are less, whereas here you can see the transmission, neurotransmission at a more higher level, and this is mainly happening because of the glutamate receptors like the NMDA and AMPA. Both the type of receptors of glutamate are playing a role in long-term potentiation. Okay, so we'll move on to the next question. Which of the following is related to fear processing? Visual cortex, hippocampus, amygdala, olfactory bulb. Which of the following is related to fear processing? Visual cortex, hippocampus, amygdala, olfactory bulb. Your time starts now. Start answering. Is related to fear processing. Visual cortex, hippocampus, amygdala, olfactory lobe. I can see only one answer. Where are the rest answers? I need answers from each one of you. I'm telling again and again. Be active in the class. Yes. Take your time, think properly, and then answer. Most are saying amygdala, some have said hippocampus. So it's one of the either hippocampus or amygdala. We'll see. Let's see what is the right answer. So the right answer is amygdala. So whoever is given amygdala as the answer, they are correct. So what is the explanation? So the question was, which of the following is related to fear processing? So amygdala is the region which is related in regulation of our fear processing. So here you can see the structure and here you can see the location of amygdala in our brain. It is near the hippocampus, but hippocampus is not involved in stress regulation in our, like, fear regulation in our body. Amygdala is the only one which is responsible for the response and memory of emotions, especially fear. We have a lot of emotions going on, but amygdala is the one which gives response for fear, right? Okay. We'll move on to the next question. Osmolarity of 1 molar sucrose solution is, you, I think you need to do a calculation. Actually not, but if you want you can do it. Osmolarity of 1 molar sucrose solution is 1 osmol per kg, 1 osmol per liter, 10 osmol per kg or 10 osmol per liter. It is a very simple question. You just need to know the formula of osmolarity. I think we have learned this in our junior classes. And let's see how many of you can answer it. This is a very simple question. Osmolarity of 1 molar sucrose solution. 1 osmol per liter, is it? I want answers from all of you. I'm seeing some names who are only answering and I can't see the names of all of you who have joined today. Yeah, whoever are joining late, please try to cope up with the questions. Start answering the questions, and whoever have missed the previous half an hour or so, they can catch it later through the YouTube session. Is everyone answering? I can see only 4-5 people answering here. There is no point in discussing if you don't answer at all, right? Definitely you will know the right answer, but it is only effective when you try to answer the question thinking of your logical reason. Either way, it's correct or not, it's okay. But if you don't answer at all, if you don't try at all, definitely you will forget after 2-3 days of our discussion, right? I need answers from everybody. Let me know if you need some more time to think and answer. I can give you more 2-3 minutes, but yeah, try to answer it. Osmolarity of 1 molar sucrose solution. Whoever is raising hand, please write your query in the chat box. I'm seeing the chat box. Whoever is raising the hand, what is the issue? Is there any issue? Is everyone done? Should I say what is the correct answer? Osmolarity. That is what you need to count or calculate for 1 molar sucrose solution. So the correct answer is 1 osmol per liter. I think how many of you have answered? They have answered it correctly. Okay. So how do we calculate osmolarity? We have already learned this from school, right? In chemistry. that molarity into n. And what is n? Number of particles that dissociate from the solute molecule. So does sucrose have any molecules? Does sucrose have any particles that dissociate? No, it doesn't have. It is a single solute particle or a single solute molecule. Here I've shown examples to make you understand. Like for example, if we take potassium chloride, it has KCl. The molecular formula is KCl. So it has two particles, K plus and Cl minus. Similarly, if we take calcium chloride, it has calcium and chloride. But if we dissociate them, if we separate them, there are three particles. One number of particle of calcium and two number of particle of chloride ions. Whereas sucrose, they doesn't dissociate into particles. They are as a whole one solute molecule. So what is the osmolarity of sucrose solution? So molarity is 1 molar. It was given in the question, and N is also 1. So your answer is 1 osmol per liter. If we count for potassium chloride, which has 2 particles, the answer will be 2 osmol per liter for 1 molar solution. Similarly, for calcium chloride, if we see, as it dissociates into 3 particles, the osmolarity will be 3 osmol per liter. I guess these examples have helped you understand how to count molarity of a osmolarity of a solute. Okay, we'll move on to the next question. Onset of Alzheimer's disease is linked with reduced synthesis of. Onset of Alzheimer's disease is linked with reduced synthesis of glutamate, GABA, epinephrine, or acetylcholine. Your time starts now. Start answering. Onset of Alzheimer's disease is linked with reduced synthesis of. I think you need to know about the disease to know this. Of course, if you have attended the lecture, you can answer it correctly. I'm not seeing many people actively answering in the class. This is not good at all. You should be active, try to answer it. No one is punishing you for giving the wrong answer, right? We are all here to learn something from this class, something effective from this 1 hour discussion. We are discussing these questions for your practice so that you can get to know more concepts, revise your concepts through these questions, right? Onset of Alzheimer's disease is linked with reduced synthesis of glutamate, GABA, epinephrine, or acetylcholine. Many of you have said GABA, some have said acetylcholine. Is everyone done? Should I say what is the right answer? I guess there are no new students in the class. Like it's already week 6, so I'm hoping that all the old students are only joining the classes, and I don't need to give. Okay, we'll see what is the correct answer. Acetylcholine is the correct answer. So few students gave the correct answer. Nice. So here you can see the normal condition brain and synaptic transmission and the Alzheimer disease signaling process. So what happens in normal conditions? The cholinergic neurons are acting here. What are they doing actually? So under normal conditions, postsynaptic resident neurons of the cortical and the hippocampal regions, here you can see from the hippocampal region, these cholinergic neurons are present, which release the pro-NGF, which is again then taken up by the presynaptic cholinergic neurons through, here you can see the receptors TRKA predominantly. So what happens in the Alzheimer's disease? These, if I'll explain in a simple way, these the signaling in the cholinergic neurons present in the cortical and the hippocampal regions are acting perfectly fine in normal conditions. Whereas in Alzheimer's disease conditions, the signaling process in the cholinergic neurons is not happening properly, due to which acetylcholine is not getting released, due to which there is a reduced synthesis of acetylcholine in the Alzheimer's disease condition neurons. So the correct answer was acetylcholine. If you want to know the whole detail, I have given the notes here. You can go through it. Okay, we'll move on to the next question. The molecule, not a precursor or metabolite, which is used as medication for Parkinson's disease. The molecule which is used as medication for Parkinson's disease is L-DOPA, ascorbic acid, acetylcholine, or melanin. L-DOPA, ascorbic acid, acetylcholine, or melanin. The molecule which is used as medication for Parkinson's disease. This is a very famous answer. If you know anything about Parkinson's disease, I think you should be knowing the name of the medicine out of all these. Yeah, yeah. Seeing the answers, I guess everyone knows the medication of L-DOPA for medication for Parkinson's disease. Okay, we'll not waste much time. We'll see how L-DOPA why L-DOPA is used in the treatment of Parkinson's disease. So the correct answer is L-DOPA. So first of all, why are we using L-DOPA? To replenish the dopamine levels in our brain, right? Parkinson's disease is mainly associated with low levels of dopamine, and the L-DOPA medication is mainly used to replenish the dopamine levels, or that L-DOPA acts as dopamine in our brain. So what is L-DOPA doing? It is converting the tyrosine in our brain into dopamine, which again is playing its role in synaptic transmission to throughout the dopamine receptors. The message is passed here, or it acts, either it replenishes the dopamine levels, that is the levodopa, that is the action of the L-DOPA, or dopamine agonists, which like mimic, they similarly act as dopamine. They are also being used for medication of Parkinson's disease. But for the answer, L-DOPA or levodopa, that is a drug that converts tyrosine in our brain to dopamine, right? And other medications are also there, like monoamine oxidase B inhibitors, which also prevent dopamine from breaking down in the brain. So in Parkinson's disease, what happens? The dopamine levels become low in our brain, they break down. So these inhibitors, these oxidase B inhibitors also prevent the dopamine from being breakdown. But the most famous treatment for Parkinson's disease is L-DOPA till date, although it has not been a permanent cure for Parkinson's disease has not been discovered yet. But whoever are suffering, people who are suffering from Parkinson's disease, they take L-DOPA as a medication, right? It is the most effective one till date. We'll move on to the next question. Sleep circadian rhythms and body temperature are all controlled by dedicated neuronal circuits in the sensory system, hippocampus, hypothalamus, or pituitary. Your time starts now. Start answering. Sleep, circadian rhythms, and body temperature are all controlled by dedicated neuronal circuits in the sensory system, hippocampus, hypothalamus, or pituitary. I need answers from everyone. Hypothalamus. Most of you are giving the answer as hypothalamus. Okay, let's see if it is the right answer. Yes, hypothalamus is the right answer. So, hypothalamus is mainly involved in our regulating our sleep cycle, circadian rhythm, the whole body time management, and as well as the body temperature, and dedicated neuronal circuits are present in the hypothalamus for controlling these functions in our body. Right, so here you can see in the figure, hypothalamus is involved in stress regulation, circadian information, it takes in all the things, as well as it regulates our hormonal levels in the body as well as nutrient levels in the body, as it plays a role in how much food we are intaking and how much energy is being produced in our body. So it has a very vast role in our body, that is the hypothalamus. Okay, we'll move on to the next question. That is, this is the last question of our session. So, dash may be released as transmitter in neuronal preparation or as a mediator of an inflammatory response by mast cells. Dash may be released as a transmitter in neuronal preparations or as a mediator of an inflammatory response by mast cells. The options are acetylcholine, glutamate, histamine, dopamine. So which one of these is released by mast cells? The question is, which one of these is released as a mediator by mast cells for an inflammatory response? I need answers from all of you. This is the last question of the session. Is everyone done? I'll give you some time. Try think, but try to answer. Try to answer. I'm not seeing everybody is answering. I need answers from each one of you. I'm seeing the name of the persons who are answering. I really appreciate whoever is answering all the 10 questions, either wrong or right, but they are trying effectively to be a part of this interactive session, right? Okay, is everyone done answering? Okay, so the correct answer is histamine. I think we all have read this in immunology as well. If you all are science students, you must be knowing this. So histamine is a chemical mediator that takes part in a allergy reaction. So whenever an allergic reactions occur in our body, also known as an anaphylactic reaction, what happens? The whenever antigens enter in our body, antibodies are secreted. Which antibody type is secreted? IgE is the antibody type which is mainly involved in allergic reactions. So when the mast cells detect these IgE antibodies, these you can see after the antigen enters in our body, the B cell gets activated and it secretes the IgE antibodies. And when these IgE antibodies bind to the mast cells, the antibodies bind to the mast cells and the antigen binds to the antibody. There is a release of histamine from the mast cells, and due to this release of histamine, there is an inflammatory reaction in our body. Obviously, whenever we get wounded by any kind of accident or something, whenever there is a wound in our body, our body, that region in our body gets inflamed, right? We all have noticed that inflammation is because of mast cell, because of the histamine released by the mast cells. Inflammation means what happens, the histamine makes the blood vessels expand and swollen, and that skin area becomes itchy because of the release of histamine. That is nothing but a anaphylactic reaction, allergic reaction that is happening. That is, the antibodies are working on the antigen that has entered in our body, right? So that is the function of the mast cells. This is also a hypersensitivity type 1 kind of reaction, anaphylactic reaction. Okay, so this was the last question. I want to know the score of each one of you. Out of 10, how much have you all scored? Supraja Rajkumar, 9 out of 10. Very good, very good. 9. I guess today's session questions were quite easy, quite direct. And yes, you all are doing a great job. Whoever are attending the sessions continuously, some names I can see who are with me from the first session, and I really appreciate all of you. Now I joined. I'm requesting all of you to please be on time. 6 to 7 every Tuesday is the timing for interactive session for the animal physiology course, right? You all have the Google Meet link with you. Please try to be on time so that we can discuss the questions effectively. Whoever has missed the session, it's nothing to worry. You can go on to the YouTube channel. I think you all have the YouTube channel link. I think NPTEL is providing the YouTube channel link. You can search the folder link. The PPT I'm uploading in the folder, and the recording of this class is in the YouTube channel. And I think you will be getting the link. Uh, the link is available in the NPTEL portal. I think, yes, it is available. But if you're asking, I'll share the link with you all. So here I am, folder link also you want. Uh, okay. Like, there is a Excel sheet available in the NPTEL portal where you can find the folder links and YouTube links of all the weeks. Till week 5, you can find them in the NPTEL portal. I'm sure it is there. You guys can go and check. Please give link on mail. I don't think it's necessary because I'm sure the links are present in the Excel sheet. Yes, I have already checked it. It's in the NPTEL site. Okay, you can see it in the home page of the NPTEL site for the animal physiology course. You can find the Excel sheet there. They have it. Okay, you just check once. If not, I'll give you all the links in the next class. Okay. So I could see scores of only 4-5 people, but there are more than 4-5 people in this class. So whoever are not active in the class, only joining and listening to the questions and explanations, I want them to please answer in the class. It will only be effective for you if you try to think and answer the questions, right? It will only help you, and it will serve the purpose for joining this NPTEL course as well. Okay, so yes, okay, so this is the end of our session. We'll meet next week, Tuesday, 6 to 7 p.m. If you want to attend the session effectively, please be on time. Then we can complete the 10 questions very effectively, right? Okay, we'll meet again next Tuesday. Thank you, all of you. Thank you. Okay, okay, we'll try to actually the problem is only with the question, only with the time, but we'll try to see how it goes. Okay, thank you.